there's all this stuff going on, and you're going to have to pan around like I am right now in order to find the very specific part that is actually the ampulla. Actually, I think I'm going to zoom out so we can find this a little faster. Ah, there we go. Okay. So, actually, I'm going to just leave it at this notation so I think we'll do all right with this. So, this entire thing, if we were just to put this up on the slide, this is the ampulla. Okay, this entire structure, ampulla. Now, the area that we're going to be focusing on and identifying parts from is this region right here. Okay, so kind of like a long-winded spiel, but I'm going to use the names as I go through it, but I'll actually go back and like identify them as I go. With the ampulla specifically, I think it's helpful if I explain kind of how it functions to go along with the different structures. So the ampulla and the crista ampullaris system, it's involved in equilibrium. So it helps you know up from down, and if you like, you've ever like been on a roller coaster or like the gravitron or any of those that kind of throw off your center of balance and your equilibrium? The reason that happens is because it moves your head around so fast that it confuses this system. So the way this works, this entire compartment is filled with fluid. This little portion here called the cupola is actually a gelatinous-like substance. It's kind of like a gummy drop. You think of it as like a gummy drop that sits on top of the crista ampullaris. And when you move, the water and fluids inside of this compartment move as well. That movement is detected by this gel, and it moves with that fluid. There are sensory cells called hair cells that sit just on top of the crista ampullaris, and they stick out into the cupola, just like that. And when the cupola moves, the longest hairs move first, and then if it's a really big rotation, you have shorter hairs on the side that will also move. So that kind of tells you like how much it's moving. So basically, when those hairs move, that sends a sensory signal to the brain that your balance is off, or like up and down are now switched. You're standing on your head. Okay? So that's kind of how the system works. And kind of a really fun, just like evolutionary fact about this too. We're not the only animals that have this system, but other animals use it in a very different way. So fish actually also have this same setup, but they have it externally. So obviously fish are in water. So instead of having a compartment of liquid that surrounds this, they have the ocean or the river that they're in surrounding them. And the reason that they don't bump into things, it's not because they have great eyesight, because they really don't. It's because they have these little sensory structures all the way down either side of their body. It's called the lateral line system. And when currents hit their body, they can detect them using this exact same system. So the cupola pick up the signal, send it to the hair cells, and that tells them that they're getting a big poof of water from that side, and they should steer away from that movement. So it's kind of a cool thing that this is seen in a lot of other species, but in fish specifically, it's used very differently. Yes?
Does this relate to vertigo?
Yeah. Yeah,
Kind of
so that vertigo is more like the nauseous feeling typically. So that's like almost like more psychological. But if you get vertigo because of like motion specifically, then that would be related to the system. Good question. All right, so I'll go over the parts now. This is the crista ampullaris, so this mountain that you see here projecting into the empty space. Just this top layer of cells at the very tip, those are the hair cells. The gel that comes off of it, that's the cupola. Okay? Some of the slides that we have don't have the best examples of cupola, but what you're looking for is a structure like this with this like almost like a projection or a villi coming into it. Okay? If you can see the cupola, great. If not, it wouldn't hurt panning around the slide to see if you can find a better example. Yeah.
So on the test, like, how would you distinguish like this hair cells? How would you?
So if we're asking for the crista ampullaris, in order to not be confusing, we're not pointing there. We're going to point here.
Okay.
If we're asking for the hair cells, we'll put a pointer right at the top of it. Okay? And then cupola is just going to be the gelatinous structure. If there's no cupola present, we're obviously not pointing to it. Okay? So don't kind of worry about that. All right, any other questions? All right, sounds good.